A very warm welcome to all our participants and attendees who have joined us today for this 24th session of our monthly series BISMiS Live. We have with us an excellent speaker today, a very young speaker in Dr. Thomas Hitch, who is working at the University Hospital at Aachen, Germany. And he'll be speaking on the topic, the world of gut microbiomes through the lens of cultivation and microbial bioinformatics. Tom has been working at Aachen since 2017. He was a bachelor at the University of the West of England in biomedical sciences, and then he switched to bioinformatics during his PhD at Ipswich University under Professor Chris Creevy on the rumen microbiome and its impact on host methane emissions. And since 2017, he's been working at Aachen at the University Hospital under Professor Thomas Clavel with his research focus still on microbial communities of the gut. But instead of ruminants, he's actually working on humans and model species for human disease research. And he's worked on multiple projects which are focused on the discovery and naming of many novel taxa, as well as the development of tools to improve our ability to describe these taxa. One of the key tool that they've developed is called the protologger. And I believe Tom is going to talk about protologger in his session. So over to you, Tom.
Thank you very much for the introduction. So yes, the title of my talk today is The World of Gut Microbiomes Through the Lenses of both Cultivation and Microbial Bioinformatics. So just to give a brief introduction into what the gut microbiota is. So within our intestines, we have viruses, prokaryotes, so bacteria, archaea, and fungi all living together. So altogether, this is termed the microbiome. However, whilst this represents around 300 to 500 bacterial species alone per individual, this differs between individuals. So I may have 300 species, you may have 300 species, but we may only share 100 species between us. Now, omics methods such as metagenomics or 16S sequencing allows us to capture around 100 to 200 species per individual. So this is only a subset of the entire diversity which exists within an individual's intestines. Similarly, cultivation allows us to capture part of this sequence diversity. So we can see the overlap here between omics and cultivation methods. And currently it's estimated we can capture about 30 to 60% of species we see via metagenomics or 16S methods via cultivation. However, we do also end up seeing that we can culture certain species which are subdominant within the original microbiome. So they are not represented within the omics method sometimes, but we can get a pure culture. So I'll go into this in a bit more detail in a minute. Now, to look at who's there and what do we know, a few years ago, we ended up doing a bit of a survey on 15,000 mouse gut samples using 16S rRNA gene amplicon sequencing. So what we have here on the x-axis is the cultured fraction. So this is the number of OTUs or operational taxonomic units, which are kind of a alternative to saying a species in 16S data. So the cultured fraction is those OTUs which matched cultured isolates. So we can see that until 2016, we had around about 30% of a given sample was already cultured, which is a good start. Now, over the next 4 years, so when we did this survey back in 2020, we were able to show that there's been a significant increase in this, but it has been slow and steady trying to get there. So now we can see that the average is more like 33% cultured. Now, when we look at who still remains uncultured. We can see that at the metagenomic assembled genome level, so MAG level, this is dominated by Lachnospiraceae, Oscillospiraceae, and Muribaculaceae, which are some of the most dominant families where we have a lot of uncultured members within the mouse gut. Now, on the human side, we see a very similar story, where in this relatively recent paper, we end up seeing that the MAGs from different phyla, which are shown on the x-axis, and the proportion of species within those phyla which are either cultured in blue, cultured but from a different source other than the human gut in red, or uncultured in green, varies dramatically across the different phyla. So this is because some phyla might be more easily able to be cultured, but also we have a difference in the number of species within each of these groups. So for example, the Bacterodota here, there's 603 species being studied there, and the majority are still uncultured, even though this is a dominant phyla within the human gut. So over the last 10 years or so, we've been working to culture many more of these species. So we've been working on a host-specific basis. So for example, we looked first at the mouse gut, and back in 2016 published the MyBC, the mouse intestinal bacterial collection. And last year we're able to publish a updated version, MyBC2, which included 212 strains, 151 species, and this included 53 new taxa, which we were able to name and are currently undergoing validation. Now, we have a track record of getting these validated because a few years prior, we did a similar process on the pig gut, where we created PyBac, the pig bacterial collection. And here we were able to name 38 novel taxa, which have all been validated now. And similarly with the chicken gut, so CHIBAC, we isolated and then named some novel taxa. And the one we're currently working on is the human intestinal bacterial collection. So in here, we have already published some papers working on these isolates, but plan to continue on in the future in this direction. And on the right-hand side here, we have a list of some of the major genera we have isolated from. Now, this has all been done mostly using traditional cultivation methods and has led to the isolation of a wide range of different bacteria. So one of our students, Alfrezel, had the wonderful idea of trying to sterile filter some feces and seeing, can we actually get any bacteria through the filter? So he took mouse feces, filtered at 0.45 micrometers, and was able to isolate this novel family called the Pumilibacteriaceae, which form these tiny cells which were able to get through the filter. Another species which we found quite interesting was Olivipapstia intestinalis, which we were able to isolate from the pig gut, but it turns out that it's actually highly dominant within the human gut, occurring within 50% of the population of the individuals we studied. And one thing we always like to look at is the functionality of these bacteria. So here we ended up isolating Fecalibacterium hominis, a new species within the genus Fecalibacterium, which is known to produce butyrate, and we have confirmed that this species also produces high levels of butyrate. Now, butyrate is interesting for us because it is associated with health and modulation of the immune system. So we would hope that this bacteria would actually be associated with healthy individuals rather than diseased individuals. And on that topic, I'll talk more in a minute. Now, whilst, as I said a second ago, we have been using traditional cultivation methods a lot, which would involve the taking of fresh feces, homogenizing it, putting it into anoxic PBS, so putting it into an anaerobic condition. similar to as it would be within our intestines. And then doing spread plate inoculation, colony picking, and then 3 times restreaking to get a pure colony. We've also been looking at other methods which might allow for us to increase the throughput of this process. And a few years ago, we were able to get our hands on one of these Cytena single cell dispensers. So the way this works in brief is that you put in your homogenized fecal sample into a chamber. It then creates individual droplets from that chamber. And if those droplets contain a single cell, it will be printed into one of these chambers in the center. So you have 80 droplets per plate that you're able to print. If the droplet instead contains 2 cells or no cells, then it's removed and put into the waste. So this allows us to hopefully get pure colonies straight from the beginning. Now, we applied this method using different media, different patients to see how many colonies we could actually capture per individual, but also, was this actually more time efficient for us to do? Would we get more isolates for the same amount of time put in? And so here, first of all, is the machine itself within one of these anaerobic chambers to ensure that there is no oxygen getting in. So it's a little bit cramped in here, but we've recently bought a larger anaerobic chamber to place it in. But on the time basis, we were able to show that the hands-on time was equivalent between the traditional spread plate inoculation method and the single cell dispensing method, as shown here in gray. And this is because when printing, you had to have someone at the machine at all times to look at the images and confirm that yes, it is working correctly. Each of the droplets does only have a single cell, because the way it detects if there's a single cell or not is that it does image recognition. It has a camera, it takes a picture of the droplet. Does it see a single cell, yes or no? So we just want to make sure that it's doing its job correctly. Now, the difference we saw was in the incubation time, because here we have a single droplet and we incubate it for 3 to 5 days. We have the pure colonies and we're able to go straight to MALDI identification, 16S sequencing, continue working on. Alternatively, with the traditional method, which took longer, is that we had to do 3 times restreaking to make sure we had pure colonies instead of a mixed colony, each one of those taking a few days. So whilst that allows us to capture more diversity, one thing we also took into account is research which has been going on, such as this paper from Ito et al. From 2018, where they've shown that different media allows you to capture an increased diversity from the same original sample. So what we have here in the center of this plot is the original feces. So this is from one donor. You have this sample here, where in color you can see the relative abundance of different taxa which were identified, and in black you have taxa which were not identified within that sample. So you can see here in the center there's a large gap where none of these bacteria are really present within the original sample, it seems. Then what they did is they tried to culture the bacteria from this original fecal sample in different media. So you can see here on the right hand side you've got BHI, PEA, and on the left hand side you've got some additional ones as well. Now what we can see is that a lot of the more standard cultivation media that we might use to culture from the human gut were able to capture this major group up here, which are all quite dominant. So this is great to see that they're all capturing similar sort of diversity there. However, if we want to use multiple media in combination, we want those which capture the most different diversity. So we might want to select one which captures this group here, one which captures this group here, and one which captures this group here. Now, what I mentioned previously in my talk about there being cultured species which don't get captured by sequencing methods is highlighted very clearly here, where all of these were inoculated with the same fecal sample. But in the original fecal sample, we can see that these bacteria here, for example, were not present, or at least were not detected using the sequencing method. However, incubation of that fecal sample in certain media leads to the bloom of subsets of bacteria, whilst, say, the dominant bacteria might go into decline, changing the profile of that sample, allowing us to capture those subdominant members. And this is shown in PEA, where we see the presence of an additional subset of bacteria, which we wouldn't have seen in the original feces. So again, this highlights the need to use different cultivation methods, to use different media. So within our work, we applied this by looking at 4 different patients, so labeled 1 to 4B, and we printed their fecal samples on 10 different plates of 3 different media. And what we were able to see is on the y-axis here is the number of non-redundant OTUs. So as we printed more plates, getting up to 800 single cells at the 10 plates, we were able to capture more and more diversity. And this occurred for each of the 3 different media we used. Now, again, are we capturing the same species here, or are we capturing different species, as shown in ITO? Now, we were able to see that a large subset of the bacteria, so up to 40% of the OTUs we captured, were present on all of the different media, and an additional 20% were captured on at least 2 different types of media. But at the worst case, we captured 40% of additional bacteria via the use of the 3 different media. So these are bacteria which were specifically isolated on only one of the 3 different media approaches. By using 3 different media to incubate an individual's feces, we're able to capture up to around 60% additional bacteria here in patient 4B. So this again highlights the need to use different cultivation methods, to try different approaches, to try different additives to the media. Now, whilst we've been using single cell dispensing as an approach, there are other papers and other groups which have been trying different ways to get to high throughput cultivation. Now, this paper from not too long ago, only a few months ago, actually uses a machine learning approach to look at images from a normal spread plate, pick the colonies, and identify which colonies are likely to be from the same species or from different species. Now, it's done by initially looking at the plate, doing image recognition and separating the colonies as shown here. It will then try to assign the colonies and cluster them based on different parameters such as size, density, and color. And this allows you to pick different bacteria all in an automated workflow. Now here, instead of using different media, they actually tried using different antibiotics, which is an interesting approach, because if you try killing off a dominant group of bacteria because they're all susceptible to tetracycline, for example, then everything else is going to be more abundant relatively to the original sample. And if you incubate this as well in different media, then you're optimizing the ability to select different bacteria, get different subsets from that original sample. Now, so far I've been talking about how to culture, how we've been applying cultivation to our work and the different environments we've been culturing from. But this gets us to the step that once we've got a collection of isolates, what do we do then? We don't want to just have bacteria and not know what to do with them. And one of the main issues is that very early on, we started coming across novel bacteria which showed no similarity with known species or very little similarity. So general process is we isolated a pure culture. First of all, we need to compare, is the isolate distinct from its closest relatives? If it's not distinct from its closest relatives, then we have a strain of a known species. That's fine. That's easy to work with. We can assign that within our culture collection and put it away. But if it is distinct from its closest relatives, Then we have a novel taxon. And this is where the hard work really begins a lot of the time, because we need to deposit it within 2 culture collections. We need to define exactly how novel that isolate is. Is it a novel species? Is it a genus? A family? A class? We need to look into these things. We also need to look at what are its functional features, either via experimental approaches or by genome analysis. And then one thing which a lot of people struggle with, myself included, is actually naming the taxon. So do you name it based on a researcher? Do you name it based on a place, on the features of that taxa? And how do you generate that name? So one thing we developed a few years ago now is a tool called Protologger. So the way Protologger works is that the user inputs their 16S rRNA gene sequence and their genome from a single isolate, and that's all the user needs to do. Everything else is automated from that point on. First of all, there is a quality check stage where the 16S sequence is checked for chimeras and checked that it's complete enough. If it isn't complete, then an error message will be sent to the user, but the analysis will continue. We then look for the closest relatives against the Silver Living Tree project, and this is filtered for living species which have a valid name and those that don't. So we specifically in Protologger study the species with valid names. So we do this against the LPSN nomenclature list, and then from the comparison of the user's 16S gene sequence to those within the Silver Living Tree project, we're able to calculate the sequence identity and create a 16S tree. We then also have a list of the 50 closest relatives, which we then use to identify the genomes of within the GTTB database. So with the genome next, we then end up looking at the completeness and contamination of the genome using CheckM. We then run it through GTTB TK, because this allows us to assign novel taxa a lot of the time, or at least see, does it share similarity with something which is novel but has been seen before in, say, a MAG. We then filter the list of closest relatives, as I said, against the valid names, and calculate ANI values, so average nucleotide identity. We calculate POCP values, so percentage of conserved proteins. We look at the percentage of GC difference between the input user's isolate and the closest relatives. And then we're able to also look at the ecology of the input species. So this is done using 2 different methods. First of all, we have a collection of environmental mags, which we compare the genome of input by the user to. So this is currently at about 200,000 mags we have, which we compare against. And secondly, we have a 16S dataset from IMNGS, which includes 15,000 samples from 15 different environments. So 1,000 samples from each of these 15 environments is included. So we can calculate the prevalence and the relative abundance of the input organism within these different environments. And in addition to this, we look at the functionality. So this includes antibiotic resistance, the ability to degrade certain carbohydrates, and we do a targeted pathway analysis. Now, this is the first version of Protologger. We're currently working on a second version of Protologger now, and hope to have that underway and online within the next year. Now, just to give a brief overview of the different taxonomic assignment methods we use. I mentioned 16S sequences, and one thing Protologger does automatically is creating a 16S rRNA gene tree. So we do the sequence comparison, we generate the phylogenetic tree for you to allow you to see where your species sits compared to its closest relatives. We also do the same on the genome scale. So this will be using phylophlan, which uses 400 marker genes, and we're able to look at all those marker genes, concatenate them together, and then hopefully create a more refined version of the 16S tree. We also, as I mentioned before, look at ANI, so average nucleotide identity, and percentage of conserved proteins. So ANI looks at the entire genome, whilst POCP looks only at the proteins, and POCP looks at the amount of proteins which match between the two organisms being compared. Now, ANI is good for being able to delineate species. So you may have heard of the 95% threshold for ANI values. If two species have an ANI value above 95%, they are likely of the same species. If it's below 95%, they likely represent different species. So in Protologger, we're able to use this to delineate species. And with POCP, we have a threshold of 50%. However, there is a gray area around this. So I encourage anyone who's using Protologger to look at the data, to actually investigate and make sure that what Protologger interprets is the reality of the situation. Because the POCP value, as I said, is 50%, but we have had cases where you may have values ranging from 47 to about 55%, and those may still represent either known genera or novel genera. Now, Protologger is freely accessible. You don't have to log on, although I do encourage it because then all of your data is saved on the system. Because we're in the EU, it follows data protection laws from Germany. So your data won't be shared with anyone else. It is stored only on the machine. We have based on the Galaxy system, so it should be pretty intuitive to use. And basically, if you log on, you'll see this homepage. If you click Protologger and then click on Protologger to run it, you'll come to this page where you then can upload your 16S sequence, your input genome, and then you just have to click execute. And after around 5 hours it takes usually to run, you'll get all of this output on the right-hand side. So you've got your matches to different mags, your GC values, your ANI values, POCP, and you will have a Protologger overview file, which kind of summarizes all of the results together and will give some basic interpretation of, do you have a novel taxa, do you not, and what functions are present, what's the ecological distribution. So we applied Protologger in our original publication of it on 3 different data sets. So what we have here is the HPC. This is a collection of human gut bacterial isolates from Forster et al. From 2019. We also have BioML, which is another human gut bacterial collection from Poyet et al. From 2019 as well. And then we've got the Hungate 1000. So this was a large collection of ruminant gut bacteria, which came out in 2018. Now, each of these collections, none of the species were validly named in the original publications. So we expected there to be quite a lot of novelty. And when we delved into it, running all of the isolates from each of these through Protologger, we were able to identify that, yes, in each of them, around 50% was known bacteria, but then the rest was novel, either at the species level, shown here in a orangey yellow, or a novel genus in green. Out of these novel taxa, we were then able to look at their distribution at the phyla level. And whilst the majority of each of these was Firmicutes, because that is the dominant phyla within the mammalian gut in general, there were other phyla which were present for which novelty was identified. So we've got some proteobacteria here, Astenobacteria here, Spirochaetus there. So we were able to look further at this. Now, this is where one of the biggest issues for myself, at least, comes of trying to name a bacteria. Now, luckily, back in 2020, Mark Palin was able to produce this tool called GAN. Now, GAN allows for the automated creation of names based on some basic input. So, for example, if you want names based around horses, you may want a middle part which revolves around gut and an end part which is microbe. Then you can get a whole list of names which are based from that. So this seemed wonderful, and I really wanted to try this out. And after discussing it a little bit, we ended up thinking, well, this could work quite nicely with the ecological output from Protologger and the functional output. So as I mentioned before, the ecological output of Protologger includes the mag analysis, where we have mags from a whole bunch of different environment, primates, rumen, mouse, ocean, permafrost. And we also have the 16S RNA ecological profiles. So here we have coral, marine, a whole bunch of different gut and human associated sites, and also things such as rhizosphere, so from plants. Now we were able to combine the 16S profile with GAN to automate this process a little bit. So here is an example. So this is a pig isolate, which we worked on a few years ago. And if we look at the plot from Protologger, we can see that in the 16S ecological analysis, this species wasn't identified within any of the different environments, apart from the pig samples, where it was present in around 35% of samples. Now we ended up running it fully through Protologger, this isolate, and we were able to identify the pathway for propionate production using these enzymes detailed here. By integrating GAN as a separate tool on the Protologger website, we were then able to make it so then this input would be used, and in total 4,096 different genus names were created, of which the GAN within Protologger ends up selecting 100 at random. And then 3 names were proposed based on the functional inputs. So the way we do this is that if you have a novel species, then only species names will be provided, and these will be based on the functionality of your isolate. Whilst if you have a novel genus, then the genus name is based on the ecology of your isolate, whilst the species name is still based on the function. So here I've got an example of one of the ecologically derived genus names. So here it is. It's quite long. We designed it so then there are 3 parts to a name to make sure that they would remain unique in the long term. Now this name actually means a microbe from the duodenum of pigs, which in this case wasn't actually appropriate because our isolate, whilst being isolated from the gut of pigs, was not from the duodenum but was from feces. So this is not a name which I would suggest using for this isolate. And so this is where, again, the manual curation comes into play. However, we were able to identify that this bacteria did actually produce propionate in experimental testing. And so this name was actually appropriate because it pertains to propanoic acid, which we predicted it produces. Now, to kind of round up my talk, I wanted to mention about synthetic communities and our work creating them. So when we have the human gut or the mouse gut, we've got a native ecosystem which is complex and has a lot of unknowns. So if we think of it like a jigsaw puzzle, we've got a lot of these gray color pieces, which we don't know who they are, what they do, what their functionality is, their impact on the host. By creating a strain collection, we're able to actually isolate all of the already known species and address things such as the novel taxa by running tools such as Protologger and then going through the process of validly naming them. So then we have a collection of known strains where we can delve into what each species does and what their impact on the host is and address these issues. Then what we can do is actually place these together to create, like a jigsaw, a minimal microbiota or a synthetic community which is modular because we can take one species out, put another one in, depending on what we want to look at. It's standardized because we know what we put in, we know the amount we put in. and we can make these available to the community. Now, we did this last year for enhancing a previously existing synthetic community called Oligo-MM12. So this was designed for the murine gut, so for the mouse gut. And we published this in Cell Host Microbe. Now, here is a phylogenetic tree of the different species we were looking at within OMM19, which is the enhanced version. And in color here, we have the additional species we included. So from the original 12 species in OMM12, we removed 2 which did not colonize well in mice. We then added an additional 9 which colonized better, and that had functionalities or features that we were after which were missing in the original synthetic community. So this was very much a expert-driven approach to design a synthetic community. So these species here, which I'm highlighting, actually were selected based on their functional repertoire. So Exibacter, for example, is a known producer of secondary bile acids. This is something which the previous synthetic community did not do. Flintibacter putridicus is a producer of butyrate, and this is something where we saw lower levels of butyrate within the gut of OMM12 mice than we wanted, so this would help increase that. And additionally, we wanted to increase the amount of fiber which was degraded within these mice to get that more similar to that of normal colonized mice. So we included Xylanibacter rodentium, which is a known fiber degrader. In addition to this, we included some species based on them being mouse-specific or enriched in mouse and being microaerophilic, so then they might colonize the small intestine better, where there is a higher concentration of oxygen. And we included some species such as Escherichia coli to expand the phylogenetic diversity, because we want to try and make the synthetic community not just functionally representative of the original ecosystem, so in this case the mouse gut, we want it to be phylogenetically comparable as well. So we want all of the main phyla to be represented. Now we then actually went and colonized some germ-free mice with either OMM12 or OMM19 to see do they colonize different gut locations, which species are present, which ones are not. So let me explain this graph in a little bit of detail. So what we've got at the bottom are each of the different species which are either present in OMM12 or OMM19, so 21 species total. In bold, at the bottom, we have the novel species which we included within, so novel in the terms of they weren't in OMM12 but are in OMM19. So these are the additional species in bold at the bottom. And now for each of these species, we have their relative abundance within different gut locations within OMM12 colonized mice. So going from light to dark is cecum, colon, small intestine, cecum, colon. And similarly in purple, we have OMM19 going small intestine, cecum, colon. So what we can see is that we've got specific colonization of many of these species. We have, for example, Adlerkreuzia colonizes in the small intestine and cecum of the OMM19 mice, but is not present in OMM12 because it wasn't placed in there. We also see that the addition of these extra species alters the relative abundance of the previously existing species, such as in Bacteroides sesamuris. Over here, we can see lower relative abundance within each of gut locations compared in OMM19 mice compared to OMM12 mice. Now, in addition to just seeing do these bacteria colonize, our interest in creating synthetic communities is to see how the gut microbiota modulates the host. Does this have an impact? What impact do these microbes have? So we were able to do mu-CT imaging on these mice which were colonized with either OMM19 or OMM12 synthetic communities, and then separate out the different sections of the mouse. So we can see the heart in red, the lungs in pink, and then different fat types in blue and green. Now, one thing we observed was differences in organ size. So we were able to see that germ-free mice have significantly smaller lungs and hearts compared to SPF. So this is conventionally colonized mice in both of these organ sizes. We also saw that OMM12 and OMM19 synthetic communities were able to recapitulate the standard conventional phenotype. And so we can see a trend of increasing size of lung across the colonization statuses. And we can see that straight away with OMM12, we recapture the conventional heart volume compared to germ-free mice. We also looked into different immune parameters. So we were able to use FACS to look at the different immune cell populations and see similarly that, for example, here in the mesenteric lymph nodes, we have in germ-free mice very few raw gamma T positive CD4 positive cells. And this is far higher in conventionally colonized mice. And these numbers increase as we go from germ-free to OMM12 to a more complex synthetic community, OMM19, to conventional mice. Now, OMM19 is a great tool for looking at mouse studies. We've got a standardized microbiota there that allows us to just look at the mouse as a unit. We don't have to consider the microbiota variability between hosts. That's already accounted for by having this standardization. However, if we want to, say, create two synthetic communities, one which is for a diseased group and one for a healthy group, then this might be a bit more tiresome to have to create each one manually using an expert sort of derived opinion, as was done with OMM19. So for this, we've actually developed a tool called Mimic2. What Mimic2 does is we input a isolate collection such as MyBC2, and we use shotgun metagenomes to provide functional profiles. Using this information, we're able to select the species which best recapitulate the functional profile of different samples. So what we have here on the right-hand side is an MDS plot where we have two different groups, susceptible and resistant. These mice were, so these are the microbiotas of mice which were either susceptible to a chemical called DSS, which induces colitis. So some mice were able to be given DSS and not have colitis occur due to the microbiota, or they were susceptible to DSS, and so colitis occurred because their microbiota weren't able to protect the host. So what we can see here is that the resistant, which is what we want, the healthy group essentially, and the susceptible or diseased group separate in their original functional profiles. And we can see that the functional profile of the predicted synthetic communities are also separating at a similar level to the original synthetic communities. So here we were creating synthetic communities of 12 members to represent each of the original samples. And what we can see here is at the top, we have the 12 species which were selected to represent the resistant synthetic community. And at the bottom, we have the 12 members which were selected to represent the susceptible animals. Now, we do have two species which are actually selected as being members of both of the communities. This is because they're all mice at this point. So there's going to be some similarity in the fact that because we're predicting the same environment, even if there is a difference in the microbiota, some similarities are going to occur. But 10 of each of the two synthetic communities were different from each other, which suggests that we do actually have different functional profiles occurring, which is again re-highlighting what we see here, which is the functional profiles. When we look at the prevalence of each of these bacteria being selected within these original samples, we can see that the resistant strains were selected more frequently in the resistant samples, and conversely, the susceptible species in the synthetic community were selected more so in the susceptible hosts. When we look at the proteins that these species contain, we're able to actually look at different metagenomic data sets, such as the IBD-MDB. So this is a cohort of patients who are either healthy or have Crohn's disease or ulcerative colitis. And we're able to look at, do the proteins within our species occur within these patients? And what we found is that many of the species which were selected as being part of the resistant or healthy group were actually containing a large number of proteins which are found significantly more in healthy controls than Crohn's disease or ulcerative colitis patients. And conversely, the synthetic community which was selected to represent the DSS colitis group, which is a model for IBD, such as Crohn's disease or ulcerative colitis, had more proteins which were identified as being frequently present in Crohn's disease or ulcerative colitis patients. So this is kind of an example of what is possible once we have a collection of isolates and once we want to start looking at the impact of the gut microbiota on the host. So I want to say a thank you to my group, in particular my professor Tom Clavel, as well as everyone else within the group, including my PhD student Jojo down here, and all of my collaborators, in particular Ahran Oran, who has helped a lot over the years with making sure that the names we propose are correctly formed and will undergo the validation process, and Jorg Overman at the DSMZ, who helps us to deposit them within culture collections. With that, thank you very much, and I'm happy to take any questions.
Thank you, Tom. Thank you for this wonderful session. We've got a lot of questions for you. Whenever you are ready.
Yep.
Okay. So the first one is from Aaron Oren. And the question is quite interesting. What are cyanobacteria doing in the human gut? In one of your slides, slide number 3.
Yes. So the chance that these are actually colonized microbes is probably quite low. But what we might have is, in that study from Almeida, they had quite a lot of samples from previously undersampled populations, so such as Madagascar and the Hadza tribe, for example, where the same hygiene levels are not kept as, say, the Western world. So these different hygiene levels may mean that different water contaminants are getting into and still being present within the fecal samples and are able to then be identified. We've seen this previously in some studies where species which are present in the water can be identified in the fecal samples. So again, this could be the just environmental factors at play.
Okay. The next question is, dear professor, what do you think is the biggest obstacle to cultivating uncultured microorganisms?
I think probably at the moment there's a lot of codependence which is present between different microbes. So we might be able to see that there's one bacteria which we want to isolate, which is highly important for us, but it only grows in a mixed culture with, say, 1 or 2 other species. Now, whilst that's okay to get a somewhat reduced community, actually being able to identify which metabolites that one species needs from the 2 other species is quite difficult and something we've looked at in the past, but haven't been able to find a good way to get around this. So for this, I'd say, continuing to use diverse different media, not limiting the imagination of students to add random things. Like we had a student add coffee into media a few years ago, which led to the isolation of 3 novel species. So yeah, trying different things always adds to new diversity, I think.
Yeah. I mean, following up from that, I see that a lot of information, especially from the metagenomics, one gets a lot of pathways which are very, very prominent that are, present in an ecosystem. Have you tried in your lab with adding these synthetic compounds or designing a synthetic media for growing these organisms?
We haven't. It's something we would like to do at some point. But I think there's quite a few labs which are years ahead of us at this point in trying to create synthetic media which kind of accurately represents the gut microbiota or something.
Mhm.
And I think there's a lot of it's a struggle, but I hope that we can get there someday.
Yeah. All right. The next question is from Father Royston. The question is Protologger containing the database for other bacteria too, or only animal gut bacteria?
Yeah, so it contains so what we have within Protologger is there is the GTTB database. So we extract the genomes which are matching close relatives of
Mhm.
your input. So this is not just host associated. This is everything within GTTB. So this will be any mags which have been reconstructed previously or genomes present within NCBI or something. Similarly, at the MAG ecological analysis, that also includes marine environments, permafrost, soil. We try to expand it as much as possible. And if anyone has additional data sets which they want included, please just send me an email and I will try to include it in the next version.
All right. The next question is, what is the sequence similarity needed to go for a new species? Below what percentage?
So in Protologger, we use the threshold. So on the 16S level, it is 98.7% sequence similarity. Below that, you have a novel species. But while 16S gives an initial idea, we find that there's not always consistency between the 16S results and the genomic results. So I would always encourage, and this is why Protologger requires the genome as well, I would always encourage the use of the genome metrics as well, because any value allows for a more robust assignment, and that in combination with the GTDB TK results and the phylogenomic tree allows you to see, okay, we predict this is a novel species, but is it really? Can we see the separation from its closest relatives, or does it actually cluster next to each other? And so it's not likely to be a real novel species.
Okay. The next one is from Akila Pignaker. The question is, can microbial bioinformatics help identify novel or uncultured gut microorganisms by analyzing metagenomic data and predicting their functional roles?
Yes. So again, this is another project which we worked on a few years ago, but had to delay for many reasons. Where what we were doing is we were using metagenomes to reconstruct mags. So we had a partial genome present. We would then try to predict the functional pathways on this genome, and then try to create media based on that. So there's a great paper from Hope Patel back in 2013, I think, where they did this for a gut microbe from the Tamar wallaby. And they were able to isolate this novel species from that using a sort of directed approach, which I think is absolutely fascinating. And I think, yes, that would be ideal if we could do this in future in an automated sort of way of we get a sample, we sequence it, reconstruct mags, create different media to isolate the different microbes, run it through an automated high throughput process, and then you've got your collection of isolates. That would be the best scenario I can imagine.
Okay. This one is from an anonymous attendee. The question is, have you done some research on bird guts? Can you give us some advice about bird gut microbiome research?
Yeah, so I mean, we did create the CHIBAC, so the chicken bacterial collection a few years ago. So I would say that for advice on this, I would contact Christian Zenner over at TUM because he is a veterinary expert. He was the lead author on this work. But from our experience, I mean, very similar to all of the other gut samples, we're able to isolate from them. I think the sample quantity is lower, so it might be harder to do the sequencing. But I think that in the same way, we just need to get more people interested in these lesser known environments to improve the databases that currently exist.
Okay. The next question is from Jay Stone. The question is, do you have any advice on the percentage of oxygen when carrying out isolation? Will oxygen improve the isolation?
So we try to do all of our isolation under strictly anaerobic conditions. So we try to keep it as low as possible. And whenever we bring anything new into our anaerobic chamber, we have to flush the input chamber 3 or so times to get the oxygen level low enough that we're happy to include it into our big chamber. But Johannes Jojo, my student, has recently found that a lot of the Allostetus species, which we've isolated from the human gut, are all microaerophilic, and so grow far better if you're growing them in a hung gate and you open up the lid just for a second and then close it again. Suddenly they'll grow far denser within 24 hours than you'd usually have to wait a few days for them to reach. So definitely there is a role of oxygen in isolating these microbes from the gut. I think using a bit of in tandem of approach, so doing anaerobic and aerobic cultivation can be quite useful. But a lot of the time when we tried aerobic cultivation, we just end up getting E. coli overgrowing the plates, which can make it a little bit more difficult.
Okay. The next one is from Tianqi Shu. Excellent. Thanks for a wonderful presentation. When isolating the gut microbes of animals, does every part of the gut, whether it is front, middle, or the back, contain the same microbes? And if not, on what basis did you choose? There are two more questions to this part. I'll take them later.
Okay, so to answer this first part, no, the different parts of the gut have different microbes. So even when, so if I go back to here in our synthetic community OMM19, we can see that Lignolactobacillus here, murinus, colonizes only in the small intestine and not in the cecum or colon of the OMM19 mice. And this is true of quite a few different species. They seem to prefer the environment of the small intestine, and then they don't really grow in the rest of the gut. Or vice versa, they might want the colon environment because that's where fiber degradation goes on. It's a bit of a slower throughput there. So they need that extra time to grow, to degrade the fiber, to then utilize it to create their own biomass.
All right. The next part of the question was, do you have any viruses involved in intestine, and what are the effects of viruses on the intestinal tract?
So there has been a recent boom in research on the gut virome. So I think one study identified 200,000 different viruses within the human gut. These vary. A lot of these are not host viruses, but are phage, so bacteriophage. So they're infecting the bacteria or the archaea within the gut. But research on this is still quite preliminary. A few have been isolated. So they're definitely there. They're likely to be quite important. but we don't know enough about them yet to make any solid claims about exactly what they're important for, I think.
Okay. Another question, last two questions. Will we see different communities in the intestine and fecal samples?
Yes. So one recent study where they actually, I think it came out last week, where they developed specific capsules which you can ingest, and then they will dissolve and open up at specific pHs and capture a gut sample. Showed that in the human gut, the different locations have different communities, and so the intestines is very different to the feces that we end up seeing. Although the rectum, it has a profile which is most similar to feces, which makes sense as that's exactly where the feces are coming from. But if you take anything more proximal, then it's going to have more and more differences to the feces.
Okay. I'll take this last question that has just popped up. What measures can we take to obtain more microorganisms as diverse as possible? Could you give some advice?
Yeah, so I mean, as I was talking about before, I think using different media, the approach in the other paper I mentioned where they used antibiotics is also an interesting one. Um, I'd say trying all of the different additives you can find. We find that rumen fluid being added to our media improves our ability to culture quite a lot of media of different bacteria. So one thing we do is we'll have our standard media such as YCFA, BHI, whatever else, and then we'll just add some rumen fluid to it, and then we end up getting a lot more bacteria. So that is one approach. Also using fecal water. So taking feces, adding a set amount of water, sterile filtering it, and then using that as an additive instead of rumen fluid, that'd also work. Because then you're getting a lot of the metabolites from the gut environment without having the bacteria there. So this is one approach I'd suggest.
Great. Well, that was a lot of inputs for our students and researchers who are working on gut microbiome. And the talk was excellent, and it was a wonderful session. Thank you, Tom. It was very enlightening. And I hope our students and participants will be benefited, immensely from your expertise.
Thank you. It was a
I'm
pleasure.
sure many of them will connect with you very soon, asking more questions. So thank you so much once again.
Yep, no worries. I thoroughly enjoyed it. And yes, please feel free to reach out if you have any more questions.
Yeah. We'll meet everybody once again on June the 17th with another excellent speaker, Dr. Marcus Goker from DSMZ. And thank you, everyone, once again for joining us, and looking forward to seeing you in the next month. Thank you.